In today's webinar, we will show you how to access and publish genetic data with SBDI. Daniel Lundin will show you how the NF Core Amplisec pipeline works for processing of metabarcoding data.
So I'm, as Anders said, Daniel Lundin at Linnaeus University. And I'm going to talk a little bit about this Amplisec pipeline that we have contributed to. And I'm also going to show, demo it a little bit at least. We'll see. It's a little bit like watching paint dry again with these demos. But so the NF Core initiative is, this is a large collaboration between different sequencing centers and other interested in getting good quality workflows or pipelines for various bioinformatic applications. And I'm going to start here with the NF Core and then go down to how it's done and to the demo and so on. And the NF Core then has this homepage here, nf-co.re. And you can go to view pipelines. And there's a large number of pipelines available here. We can search for Amplisec, so we get to what we're after today. And click that. And there's a little documentation about what this is. And as Anders said, this is for denoising raw sequence reads, so statistically correcting sequencing errors into what is supposedly the correct sequences. But it says here 1.2.0, and we're actually at the 2.0 in development now. And with the 2.0, we can do more than just denoising raw sequences. We can also do the step that Anders was talking about, where we re-annotate submitted sequences to SBDI regularly. That will be done also through Amplisec. It's just that we don't run the denoising part. We just take the sequences as they are and just rerun the taxonomical annotation step. Next, NF Core pipelines are built with a tool called Nextflow, which I'm going to describe in a minute here. But it makes them very easy to run just on the command line. So you typically run a command like this. After installing Nextflow, you just Nextflow run, and then the name of the pipeline, which is then NF Core -Amplisec. You add a profile here, which is settings for how it should be run. So what you should note here is that you don't have to download the pipeline code or anything, or keep it in a certain directory or something like that. This is done under the hood by the Nextflow tool. So this is, of course, a test command, but I'm going to show you a real command a little bit later. Yeah, here's another one. But I'm coming to that. So Nextflow is nextflow.io. It's a workflow manager, a very sophisticated one, in my opinion at least. So it can handle different computing environments. So you can run pipelines on your local computer, or you can run them on a cluster like at UpMax, or you can run things at Amazon clouds instances, and well, all sorts of architectures are supported out of the box. In addition to that, NF Core also has some institute-specific ways of running things. So UpMax is really well supported. And this is, so if we go back to the NF Core page here, this is something you give to the program here with -profile. You tell it that you should use Docker or Singularity or Institute, which could then be UpMax in our case. So what you need to run then the Amplisec pipeline is essentially an installation of Nextflow, and that is extremely simple to install. So you have these 3 steps here, which first checks that you have a correct Java version or sufficient Java version. And then you type in this command. So I forgot, but it's sort of, yeah. this is a completely command line based tool at the moment. There is something called the Nextflow Tower, which is a way of integrating a graphical interface here, but I'm only going to talk about the command line way. So here you install the Nextflow tool at your Unix command prompt, and then you can also test that it works correctly. So this is the first requirement to run Amplisec. The second requirement is that you have a way of accessing the actual software installed or software required for the different steps. And this is typically done either through Docker or that Manash talked about on Monday, which is a way of containerizing software in a sense. Or there is a tool called Singularity, which is easier to run on a cluster like Upmax because it doesn't require root access. So you can run it as a normal user. So the Upmax profile for, so this is, you give this through this profile argument to the tool, to the Nextflow tool. And the Upmax profile is using Singularity then. If you run this on your, if you want to run Amplisec on your local computer, you need to either install Docker or Singularity. In theory, Conda could be used also directly, but it doesn't work so well with the 2.0 series of Amplisec. And I think it will not be better supported either. It's actually a little bit complicated. So, two requirements: install Nextflow, install Docker or Singularity. Then we go to the command line here. So I should make this a little bit larger. I prepared a little demo directory here where I have my input reads are then in this input reads directory. So these are paired and Illumina MySec reads. As Anders said, we can do PacBio also, but I'm going to demonstrate how to run it on Illumina MySec data. So this is essentially 4 samples where you have 2 files per sample. They are also very small because this is just a test data set. In addition to that, then we have a config file. And if you want to know the parameters for an NF core pipeline, you Nextflow run NF core Amplisec. And since I want to run the development version, I give it the -rdev option here. And then I just add --help to the command. And now it will take a little while, unfortunately. So you will have to wait a little. It starts here by just logging the version of the Nextflow tool. It launches the pipeline. You see the revisions of the development version of it. And then we got a whole lot of output. I'm going to scroll back to that here. So here we see that it's an NF core pipeline and also another type of version number. And then we see the main arguments to this pipeline is the input, which can be a tab separated sample sheet. So if you have your specific sample sheets, you connect each sample name to a pair of FASTA file names, or it could be a single FASTA file or a folder containing zipped FASTQ files, so gzipped actually. What's forgotten here is actually that you can also, it's not forgotten. So the FASTA file argument here, this is what you would give it if you just want to re-annotate the taxonomy of a set of sequences, ASV sequences. Then you have to give the program the sequence for your forward primer and your reverse primer. This fourth argument here, metadata, is not something I'm going to show today, but this is because besides doing the denoising and the taxonomic assignment that we're interested in SBDI, the tool can also run a couple of ready-made analysis, like plotting the taxonomic profile and calculating alpha and beta diversity and things like that. But this is only done if you give it a metadata sheet then. So something is known about sequences or samples. Yeah, and here comes some more parameters. There's a long list here. You see that there's a PacBio and there's a single end one. PacBio is typically single end. Illumina is not. Yeah, so I'm going to skip most of them and go to. So when you run Nextflow, you can either give arguments like this. So I can give it that minus input and specify where the input is. Or I can write the configuration file. And the configuration file is, of course, in many ways simpler because the command line tend to become rather long, and the configuration file also has the strength of keeping your config, so you know what you did. It is recorded, by the way, so you don't have to from that, but you can redo it easier with your configuration file. And the configuration file then is called nextflow.config by default. You can actually have a set of that. So if you're interested, look up the Nextflow documentation for that. But here I'm just going to use this default. So Nextflow will run or read a config file called nextflow.config in the directory where you are when you start the program. So if we look into that, these are the parameters I decided I needed for this particular demo here. The project here is the UpMax or SNCC project that I'm using for the computing resources. And then we have the input, which is then a directory with files. I also need to tell Nextflow the extension of those files. And particularly, I need to say here how to differentiate between the forward and the reverse reads. So here it's a capital R followed by a 1 or a 2. Then comes the primers. Double primer equals true here means that we will look for the primer twice, because we have observed that sometimes you get artifacts where the primer is included twice in the sequence, and they will be thrown away if you run the workflow with double primer equals true. Sample inference here is, well, independent is much quicker than the other option, so that's why I chose it here. The other option, I forgot the name of that, but it collects data from all samples together instead of sample by sample wise. So the other one is perhaps better, but takes much longer. And here we can specify which taxonomical database to use for the taxonomical inference. We're working actually on a version of the GTDB. GTDB is, for those of you not in the microbe field, is sort of becoming the standard for taxonomy in bacteria and archaea. But me and Anders have been working a little bit on curating the GTDB to contain good quality 16S sequences. But we can run it just like it is. So there. And then the command becomes the Nextflow run again, and the same as before. No, Amplisec -r dev. And I just need to give it a profile. In this case, it's UpMax. So I'm logged into Rackham here. So I run it with the UpMax profile. Like that. And now it will take even longer than before. And you will also see a lot of warning messages here. And that's mostly, I hope, because we're in development phase, unless I've made a mistake now. Then we will see other warning messages as well. So all this brown here is actually warning messages that we will delete once the development version is out of development. So I can scroll back to what you would have seen here. Here you get sort of a summary of your configuration and what it will do for you. And then we wait a little bit until it actually starts. So here we see all the Nextflow different processes. So there are many steps involved in this denoising thing here. And what you see here is progress through all the steps that will be run for you. It's starting by renaming the raw data files and also formatting the taxonomy. So Nextflow has calculated that even though this looks a little bit linear, it knows that this format taxonomy is completely independent of the output of that first process here. So it starts them both in parallel. So we've actually started 5 different processes here. And if we go to another window here and just type in job info to show what we have in the Rackham queue right now. So this is the queue at Rackham at this moment. And as you can see, there are now 5 different processes waiting to be sent to a node whenever one is free. And as you can see, we have positions here around 190, so it will take a little while. But this is the way then that Nextflow knows how the UpMax Slurm type of computing cluster works. So it will submit each individual process as a single job to the queue, which means that if the cluster is not heavily used at the moment, everything will run very quickly because it will all run in parallel. Now we only had 4 samples, but you might as well have 96 samples or even up to 400 samples in a single Illumina MySec run. So this is what it will look like for a while now, and this is the watching paint dry similar that I was talking about. So since I knew this was happening, I prepared data. So we have this results is where by default the results will end up, but I renamed mine from yesterday. So we have a results pre-cooked here, which is then what the output will look like. And it looks like this. And I'm going to point out a few things here that are important. So first of all, actually, I should tell you what this pipeline info, this is a feature of NF Core Nextflow pipelines that it's very important. To become an NF Core pipeline, you must record the versions of software that was used. So a CSV file here is generated with software versions. It looks like that. And we will then run Cutadapt 3.2, Data 2, 1.18, and so on and so forth, which is excellent when you then go back and write your materials and methods for your paper. So you just go to this version and you know what was run. And it's also recorded which version of Amplisec was run. Like that. And then we have a file called overall summary that looks like this. So this is a little bit difficult to read on a terminal like this. So I opened it in as a spreadsheet here. It's actually a tab separated file. It tells you a little bit about the success of the whole process, that we started with 2,500 sequences in each of the different samples. Primers were found in about 72 to 94% of the reads. So this is an important feature also, that by default, only sequences that contain the primers are kept. The primer is deleted, but sequences that do not have a recognizable primers, they will be thrown away. And then we remove this second primer as well. We lost very few here, actually, only 3 sequences there. And then we see the statistics from the Data 2 denoising. How many sequences we had when we came into Data 2, how many survived the cutting of the length of the sequences. I forgot that a little bit, but I'm not going to go into that detail today, but it's very important how you cut your sequences in Data 2. How many pass the denoising steps? And then after the denoising is done on the forward separately and the reverse separately, and after that, the program, the workflow will try to merge them by an overlap. And here we see how many sequences survive that step. And as a last step in the denoising, we look at how many of them were not chimeric. So that's another thing that this run. And then I saw that we actually have an error here. So we get the, no, this is, no. We should have had the taxonomy here, but it's not really included the way it should. So this is statistics up until the chimera removal step. So that's a little bit about metadata about the run. We can check now again if something happened in our queue. Yeah, so we have other jobs there now. So we can see here that the renaming is done. The formatting is done, those that we were waiting for before. Now FastQC is run. So FastQ is just checking the quality profile of the reads. And cutadapt. So the first real step here is running right now, identifying the primers and removing them in the different sample files. But to go back to the output here, we have a directory called Dada2, which is where all the output from the Dada2 program is kept. And this is, Dada2 is actually used to denoise the sequences, as we have been talking about, but it's also used, so the same R library, Dada2 library, is also used for assigning taxonomy, which is a little bit confusing perhaps. But so what we can check here are the way these look. So Anders showed a typical ASV table before. This is an ASV table then in a wide format, as Anders also showed, that we have the sequence identifier here, and we have numbers or counts then for the different samples. The sequence identifier is just an MD5 sum of the sequence itself. So it's a unique identifier for the sequence itself. This is something we keep in the SBDI ASV database as well as a key. And then, well, we have a FASTA file with the sequences again. Also, I'm not going to show you that. I'm just going to show you the tax table here. Or we can take the species version actually. So taxonomy is assigned through, let's open it in Vim instead. No. Taxonomy is assigned through a so-called Bayesian algorithm down to genus level. And Bayesian here means that it actually checks the k-mer distribution of known sequences and compares that to your ASV sequences. And that goes as far down as genus in the best case. Now you see that in many cases here we have the family. Here we have actually a genus, although it's a rather technical name. This was only specified down to order level. So it depends a little bit on what the database looks like and how your ASV compares to that. And this is something like Anders emphasized, that this will improve with time. So when we rerun it, we will get more specific data here. This NA over here is actually the species assignment. And the species assignment is done by Dada2 in a very conservative way by looking only for exact matches. So you see that most of the sequences, they do not have the species. I really can't see one with the species down here in this first page. Well, here, down here, there's actually a species. So Massila and then species with this number here. Right, yeah, so we can have a look here. It's doing the same as before. And that's actually what I was going to say about the Amplisec. I hope you got the impression that this is a relatively simple tool to use, but also very powerful.